Good afternoon, everyone. I welcome you all to the second session of the day. And today we have the immense pleasure of having Dr. Soumen Naskar with us. Sir is Principal Scientist, Department of AGB, ICAR-IIAB, Ranchi. And the topic chosen by our deliberated resource person, our esteemed resource person for deliberation is gender-selected semen, a game changer for Indian dairy sector. Sir, you are audible and your screen is visible. Let's switch to PowerPoint so that your presentation becomes visible. Uh,
Yeah, sir. Before going to the presentation, just I put my video on for some moment, and then so that I can
sure,
grid
sure,
them.
no problem, no problem.
Yeah. Am I visible, sir?
Yes, sir, you are visible.
Yeah. So good afternoon, one and all attending this webinar. At the outside, I would like to thank the organizer, NC and then BIS Agriculture University for giving me this opportunity to attend this program. As I have already been introduced, my name is Soumen Naskar. I am working at Indian Institute of Agricultural Biotechnology. It's a constituent organization of Indian Council of Agricultural Research, New Delhi. Our institute works on basically two mandates. One is basic and uh, strategic research in the field of agricultural biotechnology. That is our objective one. And our objective two is to create human resources and also to do a policy support for the national level. So we have already started our postgraduate program in agricultural biotechnology, plant and genetics breeding. And one thing is that normally ICAR institutes are commodity based. So the one of the bright thing with our institute you can say is that or you can say that it's an USP for our institute is that we work at the interface of the plant, animal, fish, and microbes. So basically, I'm in the field of agricultural biotechnology, although by my training, I belong to basically a specialization in animal genetics and breeding. So now, sir, I can go to the presentation.
Yes, sir. You can turn off your camera so that, yeah, perfect. So that the bandwidth is utilized towards presentation and your audio.
Yeah. There is some issues with my network here, so I put my video. So I'm just I'm diving into my presentation, and my presentation is basically going to be around 20 slides because I know the difficulty to keep people awake post lunch. So at the first place, I don't think my presentation will go beyond 45 minutes. And then, as the organizer already told me that it's 1 hour duration. So after 45 minutes, if there is any question reserved for me, then I'll be glad to answer that. So straight into my topic. My topic is gender-selected semen, a game changer for the Indian dairy sector. And I'm going to talk to you. I've organized my talk is basically an introduction to the livestock sector in the initial few slides, followed by what are the major programs and what are the organizations involved in this sector. And third is the artificial insemination for which actually this sorted semen is meant, and directly into the sorted semen. And then what is the Indian initiative in this sex sorted semen in bovine species, and how we qualify to be here basically. So if anyone is attending who has basically a background from say veterinary science or animal science, so for him, many of the slides and many of the contents may be they are maybe already aware of. But I just got an impression from one of the attendees that majority of the participants, they are from agriculture background. So I would like to introduce to that particular field through my initial slides regarding the livestock sector. And actually we have been working in this field for, like, my team has been working in this particular field since it's almost 2017 onward. So we have started a small way, and during the span of last 4 years, so we have been able to establish a little foothold, and then we are carrying our work forward. I just go into a very brief statistics of where the livestock sector stands. I'm sure that before me, other speaker might have already told that for the last, it's more than a decade that India has been standing at the numero uno position as far as the milk production is concerned. Even if you ignore this 2019-20 figure of 198, it's basically more than 200 million ton right now, and per capita availability may be well more than 420 gram per day. Even if you take the ministry figure also, which is available, last one is up 2017-18, there also it is 184 million tons with per capita availability more than 391 grams per day. Similarly, there is a huge production of meat as far as India is concerned, and we are presently we are getting more than 3 kg per capita per year. And egg production, it has already crossed 100 billion because this figure is basically of the year 2017-18. And the proudest thing with this is that the value of output from the livestock sector at present stand at, in today's date, it stands at more than 10 lakh crore rupees. And milk is the single largest agricultural commodity, surpassing rice or any other crops. So it's a really a big thing that milk alone is contributing to such a huge margin. Means if you see milk or say dairy sector alone is contributing almost like 4% of the national GDP. So it's a very big contribution, and collectively it is more than 10 lakh crore rupees. When we talk about their standings in the world, so in the previous slide, as I have already shown,
Sir, I think the slide is not visible. Can you stop presenting and try again? Is it me or everybody else is feeling the same? Just a second. Let me turn on your microphones.
sir, I have
Yeah,
just
Everybody else, sir. Slide is not
okay.
visible.
Yes,
Yeah. There's some issue with the slide. Can you, sir? I think there's some bad bandwidth issue. Can you stop presenting and start presenting again?
okay.
I think there's some bandwidth issue with your device, sir, right now. Yeah, perfect. Now, can you start sharing again?
So can you see my screen?
No, not yet. Can you, sir, at the bottom, click on the present now option and choose the first option, the entire screen? तो अभी जैसे हाँ अभी दिख रहा है सर.
दिख रहा है.
Yeah. Let's go to basically PowerPoint. Is it visible for everyone now? तो इस तरह से है ना, इनको तो आप
yes, it's visible.
So can I continue, sir?
Please do, sir.
Yeah. So as I have been talking about, means of course this numero uno position in the milk. So that comes with a huge population of the livestock, very specifically the bovine. But if we also analyze this livestock population over the years, we will see that the actually the female population is increasing, and the male population for the indigenous one, it is like during the last 5 years at least, it has been status quo, but for the crossbred and other things, it is coming down. And we are seeing a consistent increase in the population of the goat as well as the sheep. And same is the case for the poultry. In poultry, we are seeing a stupendous growth.
Just a second, sir. Just a second. There is some issue with the slide. I am seeing it. You just need to pin at the go to the I request all the participants to go to our resource person's name in the list, and there would be a pin. You can pin the presentation if it is visible. I think it's visible. If I am able to see it, you should be able to see it also. Hello? I think it's visible.
No, sir.
Yes, sir. It is visible. Just needs to be pinned.
It is visible. It is visible. It is visible.
No, it is not visible, sir.
It is visible.
I suggest.
Not even after pin it is not visible, sir.
Not visible, sir.
Just a second.
Yes, it is visible.
No.
You do one thing.
Please again reshare it.
No, it's not. Go to the name Soumen Naskar at the in the list and pin his presentation. There would be two Soumen Naskar. Are you able to see?
No, sir.
No, are you able to see in users Soumen Naskar?
Yeah, sir.
Yeah,
There
only
would be
one.
two. There would be
No,
another
there
in
is only one.
presentation.
Only one, sir.
I may. Sir, can you
I
stop
pinned,
sharing
sir.
and can you try again, sir? There is some issue with a few of the.
Yeah.
Okay, I'll do that. So is my screen visible now?
Why is it loading in a separate?
No.
It is in the. If you check users, there would be.
Yes, it is visible, sir. It is visible.
No, it is not visible for me, sir.
You do one thing at the bottom of your. Where you see all the people in the. Those people who are not able to see. See, there is something. There would be Soumen Naskar presentation. You just need to pin it. You just need to pin it. See, let me share my screen and see. You will be able to directly see sir's presentation also. Are you? Yes, it is now. It's visible, I think.
So can I continue, sir?
Yeah, please continue now.
No, it is not visible,
No,
sir.
it is not visible, sir, for me.
I think you guys have a
pinned
network
also.
connect.
Yeah, I already pinned.
It is visible.
It is. See,
Yeah.
people are saying it is visible. It's
Pinning
not.
it, it is
It is
visible.
not visible for me, of course, sir.
You have to pin it. You have to pin it. There's an option.
Yeah,
of pin it.
I have
Sir,
already
pin
pinned.
it,
I
sir.
have
It
already
is
pinned,
not
sir.
visible. It is
Sir,
only
he-
on in mobile only.
Sir, he can leave and then join again.
Can people who are not able to see, can they leave and join again?
Okay.
Visible, please slide mode.
Yeah, go
Sir,
to
can
the slideshow.
Please
Yeah,
slide
please
mode.
go ahead.
Can you maximize the slide? Sir, can you
Please go to the slideshow, sir.
Yeah.
Yeah. Those who are not able to see, please join back again. I think there is some issue with your network or something issue with your Google Meet. Please join again. Sir, please continue.
Yeah. So if we see about the poultry sector also, we see almost the same picture, because I'll not talk about the commercial poultry, because commercial poultry is mainly controlled by the private players, and that is having fantastic growth. And most of this, what you call as a national agricultural research and education system. So we people are more concerned with the backyard poultry system, and that has also been witnessing a growth rate of almost like 50%. Although the rate of increase in the commercial poultry during the last 2 years, at least since March 2020, I'd say it has in fact come down below 4%. Now, I will also I would also like to inform you about brush you up about the developmental program that has been undertaken by the central governments. Most of us are already aware that the government of India has taken a very noble initiative of doubling farmers' income by 2022, and towards the direction during the last 4 or 5 years or so, we have seen that there has been some major programs that has been drawn. First one is Rashtriya Gakul Mission, where we have seen that the nationwide AI program has been taken up, and in the present plan also, if you see, it has been extended to all the 600 districts, where presently the artificial insemination coverage is less than 50%. So in all these places, at least 100 villages, the 100 villages have been taken up, and each of these villages, at least 200 breedable females are to be covered. Similarly, I also remember last 1 year at say 2019 and 2020, there were more there is almost like 106 aspirational districts. What we have been termed as an aspirational districts, like their growth rate has not been as per the national average. They are below national average. So a 10-point agenda was drawn up for bringing them at par with the national one. So in fact, I have been looking for this one, East Singhbhum district. And by being a coordinator for that particular district, I know that out of the 10 program agenda, there were at least 3 programs which were directly linked to this animal husbandry one. One is the artificial insemination, extensive coverage of the artificial insemination, all these districts. Number 2 was vaccination for the FMD and brucellosis. And third was related to, I think it was more related to biogas. So this way we can see that there has been a tremendous increase, tremendous focus on this AI program. Similarly, for the accelerated breed improvement program, where the main focus has been given for the indigenous breeds. So there we see like increasing their productivity, because the national average, if you see, even for the best of the defined breeds, so it's hovering around presently around 5.65 or say around 6 kg. So it is proposed that their performance has to come up to at a level of around 8 kg, and for that purpose, accelerated breed improvement programs for the defined breed has also been taken up. And under the same mission, using sex-sorted semen technology for getting assured pregnancy, that has also been taken up. So I have presented some of the clips on the right-hand side that you can see. It has been during the last year, for example, like nationwide artificial insemination program. So in the month of September 2019 to December 2019, more than almost like 7.5 lakhs AI program was taken up. Another major initiative that has been taken up during the recent past is the National Animal Disease Control Program. Many of us are aware that there is a program called INAF. So in this particular program, almost like 500,000 crore is being invested for control of 2 specific diseases. One is foot and mouth disease, the FMD. Another is brucellosis. And majority of us are aware that it is because of the foot and mouth disease we are controlling a significant part of our production. It not only it is a concern for mortality, but also the morbidity. It affects the production. And for the industry, what we have seen is that the Animal Husbandry Infrastructure Development Fund, under the Atmanirbhar Bharat Abhiyan stimulus package, which has been running for the last 3 years, that has significantly bolstered the public-private initiative as well as the private efforts. I would like to focus now on some structural changes. I would say like that has made a tremendous impact for this particular sector. Means you see that during this, I believe it is in May 2019, a separate ministry has been formed, and this ministry has been formed by carving out the Department of Animal Husbandry, Dairying and Fisheries and Fisheries. So that DASDF it used to be called. So now this DASDF has been carved out, and a separate ministry has been formed named as Ministry of Fisheries, Animal Husbandry and Dairying, with 2 different departments. One is Department of Animal Husbandry and Dairying, another is Department of Fisheries. And consequent to formation of this ministry, I believe there is no seeing back for this particular sector. And majority of us are all aware that the growth rate this particular sector is witnessing is just phenomenal during at least the last 3 to 4 years. Similarly, those who are in the discipline of say be plant breeder or say animal breeder, all of us will appreciate that a tracking mechanism is very much essential for genetic improvement programs, even for the purpose or for the sake of say disease control program. So a program called Pashu Aadhaar has been made, where each of these animal is by RFID, they are tagged, and there all the details, including the health and the productions, they are stored. So over the years, there has been a massive database that has been created, which will eventually help in disease control and more so for these genetic improvement programs. Sex-sorted semen, that's going to be our focus. That has been a major focus during the last 3 to 4 years. And as I told, this is enough through the Nakul Swasth Patra. So many of us who are already rearing livestock, they are already aware of it. And another is e-Pashu Hart. We can say like it's like a what is called the government e-marketplace and government system. It's just like otherwise called say Amazon. So similarly e-Pashu Hart. So those of us who have logged in, we can see there that you can purchase livestock, you can purchase semen, you can purchase sorted semen, you can purchase embryo, you can hire an AI technician for the training program. So these are the structural changes that have made a major impact in this particular sector. Now, as I told about the central government departments, but we know that basically this animal husbandry is a state subject. It is controlled by the state. So per se, central has a little control over it. So when we talk about the major players, it's going to be the state veterinary department, state animal husbandry department. It is nomenclatured in many different ways. In some of the state, it is called animal resources development department. Somewhere it is dairy development department, and this is also with most of the states we have a livestock development board. So again, these livestock development boards are like 2-3 different. Somewhere it is poultry development board separately. Somewhere the meat products development board is separately, means meat and egg, and livestock development board is separately. Over and above this national dairy development board, they have made the most important role, most stellar role when it comes to the Operation Flood, and that is stamped by just a statement by the World Bank, which says that Operation Flood can be viewed as a 20-year experiment confirming the rural development visions. Other than that, there are a handful number of NGOs. I'll just specifically tell their name in the sense because they have done a tremendous contribution to this particular field of artificial insemination. One is Bharatiya Agro Industries Federation in Urli Kanchan, Pune. So they have made a huge impact to this particular field. Now I will come to the artificial insemination. So it's nothing new for a student of animal science or veterinary science, but just to just for a general gathering with all kind of background. So I'll say like artificial insemination is just an alternate for the natural service where what we are doing is basically collecting the semen from the genetically superior bulls, and then we have the option to use it slightly processed, means in the form of a liquid semen, or we can extend it and we can cryopreserve it, and then we can use it. So that is artificial.
Sorry to disturb you again. I think
Yes.
there is some issue with the PPT again. Can you stop sharing and again share? I'm sorry, but there is some issue with your presentation.
So can you see my screen?
Yes, yes. Can you at the bottom say resume presenting or somewhere? Is it?
No,
Hide?
no, I have just option of stop sharing and hide. That's it.
Okay. Okay. Can you press on the hide and continue, please, sir?
Yeah. I just resume my talk. As far as artificial insemination is concerned, I will say that it has played the most important role when you can say like these genetic improvement programs of the livestock sector. If we need to point out that if there is a single technology that has actually resulted in all these changes, which is at the cornerstone for all these changes, it has got to be the artificial insemination. And the artificial insemination, it has a history of almost like it's more than now 240 year plus. It started with Splangiani in Italy, and then from there to many different parts of the country. But if we see in the Indian conditions, so one Dr. P. Bhattacharya at ICAR-IIAB, Indian Veterinary Research Institute in 1960s, they played an important role. But of course, before that, there was attempt at the Royal Dairy Farm at the Mysuru and other places. But the first organized effort started there, and since then it has distributed to all parts. So almost majority of the Indian states, wherever there is an important thing, so they have standardized and it is already in practice. So as far as the physiology is concerned, we know that on an average, an animal, if we go for a natural service, so it can just further say around 100 of daughters or say 100 of cubs. But if we take the help of this artificial insemination, so what we are doing is that one ejaculation is now it is going for hundreds of, basically generating hundreds of cubs. Just to say, for example, if the average ejaculation size is say around 5 ml average, and you have around 3 billion cells, what is actually required is around 20 million cells for a fertilizations. So you can just divide this 3 billion cells by 20 million, so you can understand So that makes the difference. And as far as the physiology is concerned, it's going to be altogether a different topic. So basically, that's an important decision where the animal shall be in the standing heat. That's an important consideration. And the bull shall be free from all genetic defects. And it shall have a good score to be, and also means it shall be progeny tested so that it comes into the pictures. And when it comes to the advantage, you can imagine that a bull which is in USA, for example, Holstein Friesian or say Jersey, but we are using the Semen here. And same is the case, we are taking Semen from here, Gir, and that can be used in Brazil or somewhere else. So it has circumvented the need of physical presence, and also sometimes the size is very intimidating. For example, the Holstein Friesian is very large as compared to our native cows. So they do not or may not allow the bull to mount. And also the risk of transmissions of many diseases. So they all are bypassed by the use of artificial inseminations. And as we all of us know that we say that the bull contributes actually more than half of the herd. And by that virtue, since a bull leaves many more daughters to the herd, so it is obvious that the best of the bulls have to be selected, and for that purpose we employ progeny testing program. And progeny testing is a very time consuming, and it requires lots of infrastructure, lots of investment. One bull is going to be evaluated by the end of 6.5 years on an average, if you don't otherwise use these genomic selection tools and say otherwise assisted reproductive technology. So since the bull is going to be evaluated by the performance of its basically daughter, and we need to have many numbers of daughters to evaluate a bull, so artificial insemination is an invaluable tool. For Indian condition, actually herd size is a big challenge in a progeny testing program, and that's why we see that we associate multiple herd along with the in limited number of attempts of this ONBS. So we understand that for this particular sector, genetic improvement is very, very crucial. And we know that almost like 2/3 or exceeding 2/3 of our cattle population is basically non-descript. So we have the opportunity to upgrade them by 3 different ways. One is when it is non-descript, we are sure that it is a non-descript, or say otherwise, we need to confirm that they are not under-characterized, but they are non-descript, so we can go for upgrading. And that decision will depend on the agroclimatic region and the other resources available. Where every resources is available, you have every opportunity to go up, then you can use the exotic breed. Otherwise, it makes sense to make use of our own indigenous breeds, which are genetically superior, like Sahiwal, Red Sindhi, Gir, Tharparkar. Second is selective breeding, where it is a defined breed. You can make use of the genetically superior male of the same breed. And next is the crossbreeding, where each one of us is aware of this, where 2 different breeds are crossed. And what we basically expect is that the progeny shall perform better than the poor parent, means of the relatively poor parent, and in some cases it shall be more than the average of both the parents. Since the second plan, we can say, like we have seen that there has been a focus on the crossbreeding program, and no one can deny, irrespective of present standing, that the crossbreed and the crossbreeding program, they have played the most important role as far as the white revolution is concerned. Since the time of independence, since 1950s, if we consider, the milk production has increased by say 10 times, and the numerical position during the last 1 decade, invariably it has been possible by the very stellar contribution of the crossbreeding program. And we have seen use of temperate breed like Holstein Friesian and Jersey, but gradually during this last 10 years, when you have seen that the multiple biotic as well as the abiotic stress factors, when we play vis-a-vis, means when you compare the 2 different this genetic background neutrally, we see that actually the genetically superior indigenous breeds, they also shall be given an equal opportunity, and thus the focus has shifted to the selective breed and also for the non-descript. Now I'll come to the state of affair for the artificial inseminations. So we have more than 50 frozen semen stations in the country, And right now, out of this 50 Fusion Semen stations, we have around 10 Semen stations which are basically graded A++, and rest are like A. And that is a concern that we do not have many A plus Semen station. Just a minute. Yes. And then if we see the production, say that lies with the NDDB like Sabarmati Ashram Goshala, then one in Salon, Rai Bareli, and then also with the BEF that is in Urli Kanchan, Pune. And also there are some A plus Semen station like in the state of Gujarat, there are 2 Semen stations which are rated as A plus. There are 2 Semen stations in West Bengal which are rated as A plus. And otherwise, most of them are in say A or B plus, like that way. So actually, this has come after 2004 when it was felt that there shall be a minimum standard for the Fusion Semen production. So since 2004, there is a rating agency, and every second year, there's all the Semen stations, they are evaluated, and based on that, the Semen stations are rated. But regrettably, even after these many years, and even if I have said that if we need to pick up just a single technology that has really transformed this sector, that it is artificial insemination only, still the coverage of the artificial insemination, it has not gone beyond, say, 30 to 35%. And what the present government actually intends to do is that to increase it above, say, 50% and above. So that's going to be a very it's a so far it's at the dream stage, we can say, although there are matching efforts, but it's going to take time. So there are multitude reasons of the low coverage, but the most important being the maintenance of the cold chain. Maintenance of the cold chain is one. Second is the extension services, below par extension services, because we are unable to take it to the farmer's doorstep. And third, also equally responsible is the very low level of awareness. Maybe so that it has not been the kind of, the extension services has not been so to make an awareness level. When it comes to the animal part, we see that the conception rate is also pretty poor. Means what we expect is that it shall be around, say, at least 60%, but that's not the case. And there are a multitude of reasons, but mostly it is a nutritional, heavy loads of parasites, etc. One thing obviously comes, just like human field also, or any other field, that we collectively call this artificial insemination, multiple ovulation, embryo transfer, assisted reproductive technology. All this come under assisted reproductive technology, in vitro fertilization. All this come under assisted reproductive technology. Now, whether it has a side effect. So over the years, the data generated, at least in animal science, I will say, is the most robust across all species. We know that this artificial insemination, it does not have any significant this deleterious effect. We used to hear about the large head syndrome and others, but they are not anything. There is nothing significant there. When it comes to the production and reproduction on the female part, obviously there is nothing wrong with the at least artificial insemination. Now I will come to the very specific topic where actually these are all the before slides, they have been mostly related to introducing you to this particular field, why this is important. Now I'm going to come to the very specific topic, what I'm where I also include our own work. So all of us, we now hear this sex-sorted semen. It's quite a fancy word these days, I'll say. But the history of this sex-sorted semen is not so new. It actually a lab of USDA Bell's Philip. So in 1980s, it was first reported using this flow cytometry. So before that also, there have been many different attempts and many different techniques which are used by many people, including us. In our lab also, we do the same thing, that we use the modified swim-up approach. we use the differential centrifugation approach. But to be very frank and honest, modified swim-up differential centrifugations, they all generate non-significant results. They are inconsistent and they are unreliable. There is no significant level of enrichment which is possible by this technique. Some of the labs, they have made the electrophoretic separation, but again, that is also, it doesn't generate a good results. Although there are almost like 10 different techniques to separate, say, X and Y, but at the end, the problem is not by just separating X and Y. The problem is the cell that you take from me, it has to be returned in the same condition, if not better. So there lies the major challenge. And by this time, we understand that almost 9 out of the 10 techniques, practically their result is not robust. They are unable to discriminate X and Y significantly. Even if they can reasonably segregate X and Y, means anything above 50-50. So the sperm, when you use it for in vivo use, so that is not fit for the purpose. And by this time also, we also understand physiologically, if we see the sperm physiology, we understand that practically there is no differences between the X and Y cell in order to discriminate them. So the only viable technology, only viable technique by which that X and Y cell can be made separated is basically by flow cytometry, and that's also the only commercially viable technique. And how does it work? It basically works on the basis of differences in the DNA content. Across the species, if we see, there is a difference in the DNA content in between 2.5 to 4%. We know that the female means it basically contains 2 X chromosome, and when it comes to the male, there is a 1 X and 1 Y. Now, on an average, an X chromosome, it contains 2.5 to 4% more DNA than a Y chromosome. It varies from species to species, and it also varies from breed also. Yeah, there is a variation at the breed level also. That's very interesting. So if we compare, for example, like temperate breeds and Indicus breed, there is a difference. Even, for example, when you compare the Sahiwal with Tharparkar, we see a difference in the DNA content between the X and Y chromosome. So on the right, there is you can see the 2 pictures. So basically this flow cytometry machine, but this is not a normal flow cytometry, means outrightly you cannot just make use of a flow cytometry for the purpose of separation. You can experimentally produce, but it's not going to harvest you something which is noteworthy, because it has multiple modification, and this machine harvests more than 30 numbers of patents in terms of modifications. So as I told, it was basically generated in Texas A&M University, USDA, and initially this technology was transferred to XY technology, which is presently called the sexing technology. So XY Tech, they were the sole proprietor, and then because of the trade monopoly, there was a legal action that was initiated against them, and this because of this trade monopoly, their operation was restricted by the March 2018, and they were told that it will not be possible beyond this August 2018. So thereby the trade monopoly of the sole proprietor of this flow cytometry based separation of the X and Y chromosome, that has ended. And since then, what has happened is that the sexing technology, or say XY technology, they used to have many numbers of bull stud farms, for example, like Genus, ABS and others. So over the years, they have built up their, you can say, like reversing of the technique, and they have been successful in standardizing their own, or just like what we do in our own pharma, like you have one is product patent, another is process patent. So we have the option to change a little bit. So they have standardized their own technology, and they are also able to separate this X and Y. And with the closure of this trade monopoly, what we have seen in the Indian condition, that very quick collaboration has been forced. For example, like sexing technology, which used to be the sole proprietor. So we have seen a collaboration of BAF, Urli Kanchan, Pune with the sexing technology. And we have seen the ABS genus, which is a renowned superior genetics company for the bovines. So they have collaborated with Chitale Dairy Farm in Maharashtra also. So now this sex-sorted semen is available commercially. What used to be previously imported and exclusively would be of the temperate breeds, for example, Sahiwal, this Holstein Friesian or Jersey. So now we have the option of many different indigenous breed, almost more than 6 different indigenous cattle breed: Sahiwal, Gir, Tharparkar, Haryana. So all this breed, and similarly for the case of buffalo also, Mura, Jafrawadi, Mehsana, Bhadawadi. So we are able to get the sorted semen, sex sorted semen. And other than these 2 private organizations who actually pioneered this sorted semen technology by quick collaboration with the MNCs abroad, there have been, as I told earlier, that this Rashtriya Gakul Mission, RGM. So initially the top 10 semen station, they were picked up based on their basically rating. So subsequently what we have seen is that 2 semen station has been dropped because it was not progressing well. And then during the last 1 year, we have seen that another 2 semen stations, they have been incorporated. So presently it goes up to 12 semen stations. And commercially already, there are more than 2 states from where we are able to get commercially sex-sorted semen. One is Uttarakhand Livestock Development Board in Rishikesh. They have actually pioneered since end of 2019 and beginning of 2020. They are commercially giving. Same is the case with the Maharashtra. And what we are going to see in very near future, means it's going to be more than half a dozen of the state. They shall be able to deliver the sex-sorted semen to the field units. Now, there are other alternate technology also. We have seen that in recent past that has come up. It has come from South Korea. There is one farm called work on the principle of the agglutination. So where what is done is that basically the means once the semen straw is made out of the liquid nitrogen, so then it is thawed at the 37 degrees centigrade for 1 minute, and then it is used for the artificial insemination. Here what it is done that using the principle of the agglutination, this straw is put there, and then one has to wait at 37 degrees centigrade for 30 minutes, and then again by the capillary action, then it is reverted back to the straw. It is used for the artificial insemination purposes. This is not something I believe. This is a technology, and of course, this can be practiced almost at least in a private heart, large side heart, or maybe institutional heart. So here you can see, like in traditionally, we have around 50% sexing ratio. So if you use 10, so it's going to be 5 male, 5 female. So next generation, you are going to get 2.5 from this 5 female. You are going to get around another 2.5 female, and next generation 1.25 female. So this way, basically, you end up by the generation F3, you are going to get 10 plus 10. So this way 10, then 5, and then 2.5. And then this way you are going to get 18.75. Whereas if you are using this whole mom semen, it is for the eggs. So you are going to get, provided it has a sexing ratio of 90%, means out of the sorted semen, more than 90% are thoroughly screened. So in that case, if you are using it for the 10 female mothers, so you are going to get 9 heifers. And from the 9 heifers, again, you just take the sorting efficiency of 90%, so you are going to get 8 female. So by the end of F3, you are able to get like out of 37 total numbers, you are going to get 34 female. So there lies the this particular principle, which significantly improved this one. But it is in my personal opinion that it is not something which is really going to work in case of in Indian condition. Now, what has been the initiative in Indian conditions? We know that by the 12th 5-year plan, this National Dairy Research Institute and Anand Agriculture University, they were given this mandate for generating this technology. Until 2017, I don't think we have heard much. And then 2017-20 period also, there have been, of course, there might have been some progress, but we didn't see any commercial product. During the year 2021, again, there is a incentivization of agricultural research, where we have seen a large-scale research program has been undertaken by ICAR, roping in the few of the CSIR organizations, where they are going to employ an development of an alternate technology for this separation of the X and Y cell. And that's going to use, including the other means also, it will also include microfluidics. By the end of 2021, we have heard that NDDB Dairy Services, NDS, they in collaboration with the Jiva Science in Bangalore, so they have come out with this product, which is basically work on the principle of the microfluidics. And for this work, there is a collaboration of NCBS, Indian Institute of Science, and IIT Madras. But I'm not sure whether this product is available in the market, although it was rolled out maybe by August 2021. But I don't know whether this product is commercially available now. Now I will come to what we have done. Is that I cannot say that we have been directly trying over these last 3-4 years over generating a technology per se, but what we have been doing is basically trying to find ideal candidate which can be the basis for development of something, development of an alternate something if possible. And so we basically try to understand the differences between the X and Y spermatozoa of indigenous cattle. And as I told you earlier, when you talk in terms of the physiology or say biochemistry per se, there is no difference in X and Y except for their differences in the DNA content. Now there is differences in DNA content, so obviously there is going to be differences in gene. There is going to be differences in the protein also. Now, for example, the present technology which works on the basis of flow cytometry, so there what is done is a particular dye is used, hex 33342, and since the X chromosome on an average contains say 2.5 to 4% of extra DNA, so the fluorescence generated is higher in case of X sorted sperm, X spermatozoa bearing X chromosome as compared to Y. So otherwise, this is invasive. Invasive in the sense, although hex 33342 is a fantastic dye, otherwise it does not cause any other effect, but it has to bind to the nucleic acid, this nuclear materials, nucleic acid. We ideally believe that the target molecule shall be on the surface. We shall not get into, because it is a highly delicate cell, it's a germ cell, and it is something which is going to give rise to the life, the next generation. So any damage done here, so more than 99% cases it is going to be rejected because it will not be able to fertilize. And even if it fertilize, say the remaining 1% of cases, so what we are going to see is that we can see it can, even if it is fertilized, it will not develop into term. Even if it develop into terms, there may be some effects also. So we believe that the target molecule shall ideally be on the surface, that is on the sperm membrane surface. And we took up a bottom approach, and in the bottom up approach, we focused on the membrane proteins. And now those who work in the field of proteins, they know that means they can also endorse my view that membrane proteins are not an easy target because they are hydrophobic, and being hydrophobic, you cannot typically study by like in gel approach. In gel approach, what we do, like we do polyacrylamide gel electrophoresis. We go for 2-dimensional gel electrophoresis, and then we go for other technique like Western blot, so that you can identify the different proteins. In our case, we tried also in gel approach, but in gel approach was not working. So we went for a in sol approach. So like trypsin and lysid digestions, and followed by this LC-MS. And we identified more than 1200 numbers of total proteins in case of spermatozoa, and more than 470 numbers of proteins. So then what we did is that we did a number of bioinformatic analysis, and in this process, basically we have come out with the 2 outcomes. First is we have developed a protocol, we have developed a method by which you can make a significant enrichment of the spermatozoal membrane, because spermatozoal membrane is going to be the primary material for your research work. So we have developed a strategy, we have developed a method which has been validated by our own analysis, that the spermatozoal, this membrane protein is more than 57% by applying our method as compared to what we have seen in the others. That is number one. And number two is that the proteins that we have isolated, then we have through different bioinformatic analysis, we have classified them for their functional and the biological classifications. Means what proportion of this binding is actually meant for binding activity, how much of them is for the catalytic activity. So you can see, like when you go for the functional capacity, this functional analysis classification, so you see that a significant part actually goes for the binding proteins. And next comes is the catalytic activity, and so on, transcription regulator and like. Similarly, when you go for the biological classifications, we see like majority of the proteins are involved in the cellular process, some are in the biological regulations, then some are in cellular different component organizations and metabolic processes and so on. And finally comes our main revelation is that we have been able to identify more than a dozen proteins which are different between X and Y. So there is one protein which is called disintegrin and metalloproteinase domain containing protein 1b and KRT4, which are actually differentially expressed between X and Y chromosome. Whereas we have identified another 12 protein, what you see is that there are 4 proteins which are present in X but not in Y. For example, like transmembrane protein 190, keratin type 1 cytoskeletal protein, desmoplankin and keratin cytoskeletal 5. All these proteins, they are mostly involved in the structural integrity of the spermatozoal cells. So these proteins are, we could find them in X, but not in Y. And similarly, we found almost 8 numbers of different 7 proteins. For example, elongation factor, seminal plasma protein, this is quite a common protein which was earlier also reported, like 30 kD protein 1. And then leucine-rich repeat and fibronectin type protein. And then voltage-dependent anion selective channel protein. So this way we have identified a host of candidates which are present in Y but not in X. Now, interestingly, the proteins that are present, we found that present in X but not in Y, they are related to the structural integrity or the structural stability of the cell. So that means somehow it gives the impression what was earlier known, that the X chromosome is structurally maybe a little more robust. And when it comes to the Y protein that we found in Y but not in X, and these proteins are actually candidates which are related to the acrosomal integrity and as well as the motility. So some way this confirms the earlier belief that maybe Y chromosome swims faster than the X chromosome. And in fact, that is also the basis of the microfluidics. So this has been one work that we take pride that we have been able to do this with our limited facility, and we have been a starter. So we have published that in 2021. And then actually our purpose is going for a candidate gene approach, where we want to identify specific gene, and so that they become candidate for say genome editing or alternate. And we are also exploring nanotechnological approaches. So by this time when I am delivering this talk, we are not in a position to share that result until the time we come out publicly with this one. But prima facie, we have got something. We are getting some encouraging results using nanotechnological approaches. We are able to bias the cell population, but it requires a validation in the sense, because the cells that come out, that has to be used. Means that has to be used for this at least in vitro fertilization, and preferably in vivo to see their use. So with this, I will conclude that this sex-sorted semen, it is basically a disruptive technology, and that's the need of the hour. If we want to go into the next phase of development in this particular sector, then definitely sex-sorted semen is the disruptive technology which is able to cater to the demand of this particular sector. It will invariably increase the milk production scenario it will definitely contribute to the noble goal of doubling of the farmers' income. And the way during the last 2 years, the number of startups, the number of mechanization, the degree of mechanization that we have seen in the dairy sector, we can definitely expect that it's going to get a boost with this disruptive technology of sex-sorted semen. I would like to acknowledge my funding agency, which is my talk. Our work has been sponsored by, basically funded by this Science and Engineering Research Board of Department of Science and Technology, as well as my institute, Indian Institute of Agricultural Biotechnology. As far as the raw material is concerned, we have got support from the West Bengal Livestock Development Board and Bharatiya Agro Industries Federation. We also got support from one Indian Institute of Natural Reasons and GAM located at Ranchi. And then one Dr. Lakshmi Vandana Rongalash, who worked as again, SCRV funded National Postdoctoral Fellow. She did a fantastic work as far as this work is concerned. And Dr. Imran, he has also contributed very significantly. So thank you very much. Thank you for your kind attention. It's to the moderator, sir.
Now, if there are any questions, please ask in the chat, or you can turn on your microphones and ask.
Sir, what is presently the cost per citra of this sex-sorted semen?
Sir, if I could hear your query, your query is, what is the cost of sex-sorted semen?
Yes, sir, per citra.
Yes. So, sir, if you say the average price as it stands, if you see the private sector, so it is costing around 1,500 rupees. And the major cost, it's basically being a proprietary technology. So bulk of this is basically for the for the patent basically, and also the establishment cost, because you need to establish a flow cytometry facility. So it's going to almost cost you nearly 2.5 to 4 crore facility is going to be. So that way the cost is higher. But if you upscale it significantly, then it is possible to come down. Because what you see is that like Uttarakhand Livestock Development Board, as I told you, they have been commercially selling this product since last year, 2020, not 2020, end of 2019. So they have been selling it, and they have been selling it, to my knowledge, it is around 900. Some other states, they are selling it in 850 rupees. Some states are selling it 1250 rupees, because the government has different level of subsidy for this. Like Uttarakhand state, they have already declared that their target is basically 100 rupees. So at present, a straw of unsorted semen costs you 20 rupees. And what they told is that this sex-sorted semen, they are going to sell it by 100 rupees. So this way, sir, if I could answer your query, as far as the cost is concerned, what we are seeing is a gradual decline in price from 1,500 rupees to 100 price. And I'm sure that I'm hopeful that by 25, it shall definitely reach 100 rupees per straw. And this 100 rupees is going to, yeah.
Yes, sir. 100 rupees is affordable because by the farmers, and because 100 rupees means around 300 rupees. Keeping into view the success rate of artificial insemination, it means almost around 200 to 300 rupees, even if it goes 100.
Yes, sir. Yes, sir.
At present, if it is 900 as in some state, that means it is around 2,000 to 2,700 cost for a farmer. So at field level, at present, it is difficult for the farmers unless some government comes with some heavy subsidy on sex-sorted semen.
Right, and that is highly expected in the sense, because there are more than almost like a dozen numbers of semen production facility, sex-sorted semen production facility from the state government organization itself are coming up along with the private sector. Since there are private sector, so there is going to be a very tough competition. So people has to keep the price very competitive. And second is, I'll definitely say that you do not look at the cost of a single straw, because just imagine if you're going to get an extra female just by using, say, few amount of rupees extra, and that is going to give you one extra female calf. So basically, over a time, you are going to get more than ₹1,00,000 by investing few hundred rupees extra.
Yes, sir. If it is margin is few hundred rupees, then it is okay. But if it is say margin is by 700, 800, 900 rupees, then commercial dairy farmers may opt for it. But other dairy farmers who have just one or two cows at home, which usually is the scenario, then it is difficult
Yes.
for them.
Right. Right.
Thank you, sir.
Thank you. Thank you very much.
Any other question, please ask.
Hello. Sir, I have a question. I heard
Yes,
that
sir, please.
the success rate or the conception rate for the sex sorted semen is less. Do you have any suggestions on that or any comments on that?
Sir, that's a very good question, sir. Sir, you see the success rate, what I will say as a fertility rate. So fertility rate of unsorted semen and sorted semen, it does not differ significantly. Even in the state where I am presently working, Jharkhand, here for the last exceeding 3 years, a very large scale study has been taken by BAF, Bharatiya Agri Industries Federation. And same has been the case in the state of Maharashtra. Same has been the case in state of Tamil Nadu also. So it has been found that there is no significant difference in the fertility. And I will say now the sorting efficiency is actually more than 90%. Sorting efficiency is more than 90%. And there is a very robust result, robust field result based on experiments by both government organization as well as the large scale NGOs involved in this particular sector during the last 3 years, that there is no significant difference in the conception rate by using this sorted semen and this unsorted semen. So success rate is almost at par.
So that means the challenge lies with the technique of artificial insemination mainly in that case.
So I don't know, means how I don't think I'll be able to, I can't endorse this question, because you see, artificial insemination per se is a very proven technology, sir. It's as I told, it's more than 240 years it's going to be. And even in this country, starting 1960s, it has been being used.
No, sir, we don't have a question on the technology itself, but I'm talking about the technicians using them. I'm employing them in the field.
Sir. But one thing I definitely tell you, sir, that this is the advantage when the new ministry has come up. Even the payment method, sir, if you see during the year 2021 itself, if you see the website of Minister of Animal Husbandry, Dairying and Fisheries, Minister of Fisheries, will make the payment in batches. So the payment is not made at one go. The payment is made that once it is inseminated, a part of the payment is released. Once conception is confirmed, a part of the payment is released. Once post-conception, once the delivery is made, and you get that desired skewed sex ratio, then another batch of payment is made. Same is the case for the most of the private companies. They are asking that please retain the straw that is being used. I have attended many presentations by these sexing technologies, BAF, and others. So I see, what I see is that a very high degree of assurance, because this technology, I will say that it's really standardized. It's very robust right now. And this periodic phase of payment actually confirms that there is not much maneuverability to deliver a poor result actually. At every stage, there is a positive pressure. Unlike the unsorted semen, here there is a positive pressure that it has got to work. So I believe, sir, it is going to work. So per se, it's not going the artificial insemination. I'll more say the artificial inseminator is going to take special care, because his payment is directly linked to the success of this insemination.
Okay, sir. Thank you. Thank you for the answer.
Thank you, sir.
Hello, sir.
Yes, sir, please.
Whether the sorted semen is for only in cow or it is also present in buffalo in Maharashtra, sir.
Sir, it is present for cattle. It is present for buffalo, both, sir. If you are talking from Maharashtra, so there are two organizations which are giving you, three organizations which are giving you sex sorted semen. One is BAF.
Yeah.
So BAF is giving you semen.
Yes.
Urlikanchan. Second is Chitala Dairy Farm. So
Okay.
Chitala Dairy Farm, from there you are going to get this ABS semen. And third is Maharashtra State Livestock Development Board. So they are one of the semen stations. It is also, I believe it is in Pune. So it has been taken up. So again, it is in the XY technology only. So there are three organizations from you are getting, and you can get it both for cattle as well as you can get it for buffalo. You can get at least 6 different breeds of buffalo in the state of Maharashtra itself. You can get it from Murrah, you can get it for Nili Ravi, you can get it for Jafrawadi, and then there are another 3. So there are total 6 different breeds of buffalo. You can get sex sorted semen.
Thank you, sir.
Yes, sir. Thank you.
Sir, I have a question.
Yes,
So
sir,
which
please.
are the companies for the sex sorted semen for the desi cattle like Sahiwal, Gir and in Rajasthan area?
Sir, as I told, sir, I'm glad to answer you. You're from Rajasthan, sir. Actually, if we see during the last 3-4 years, 5 states are they are making tremendous progress. Rajasthan comes actually right now second, only after Uttar Pradesh in terms of growth rate, which is
Yes, sir.
exceeding. Which is exceeding UP. It is exceeding at the rate of 35% for the dairy sector, and for Rajasthan also it is more than 21%. Sir, you can get commercially, you can get from BAF, you can get commercially from the ABS genus. So
Okay.
these two commercial farm, it is already there. And to my understanding, so there are 12, as I told, there are 12 semen stations where actually the government of India, this ministry is establishing that facility. So Rajasthan is one of them, sir. Rajasthan is one of them. So very soon you can expect that even it will from the government sector also it is going to come, sir. And until that time, sir, most of the state government, they are purchasing outrightly and making it available to the farmers. But in that case, it is not on a very large scale. So to be very specific to answer you, sir, you shall, if BAF is there in your area, sir, I'm not sure whether BAF is working in Rajasthan, Bharatiya Agro Industries Federation. So
ABS
if
is
BAF
working, sir.
okay, so if it is working there, sir, definitely you are going to get from BAF. And similarly, sir, ABS, you can just Google ABS genus, sir. So they have distributor everywhere. So I'm sure that you will get a distributor either in Jaipur or you will get in Udaipur. It will be there, sir. Or Bikaner, it will be there.
Okay, sir.
Sir.
Thank you, sir.
Any other question, please ask. If there are no further questions, then we can consider the session to be over. On behalf of the organizing committee as well as on behalf of the participants, sir, thank you. We express our heartfelt gratitude to you.
Thank you very much for the opportunity, and thank you very much to all the participants for hearing me. Thank you,
Thank
sir.
you, everyone. Thank you for joining. Thanks a lot.